QIAGEN.
Sample to inside.
At the start of the PCR cycle, increasing temperature leads to the denaturation of the DNA template, causing it to become single-stranded. A temperature decrease allows primers to start annealing to the single-stranded DNA template. How does DNA behave in solution without the influence of ions? The DNA template and primer molecule, indicated here with a minus sign, are negatively charged and therefore show less tendency to anneal to each other due to electrorepulsive forces. A push-off effect between the DNA template and primer molecule is the result. QIAGEN has developed a PCR buffer with a unique dual cationic formulation. It consists of a balanced combination of K+ and NH4+ cations and has a strong influence on PCR performance. How does the dual cationic formulation work? The positively charged K+ ion is attracted to the negatively charged phosphate groups of the DNA backbone of the template and primer. The charges are cancelled out. The K+ ion weakens the electrorepulsive forces between the DNA template and primer molecule. The push-off effect is overcome, and the primer-template annealing complexes are stabilized. The stabilizing effect of K+ does not distinguish between correct and incorrect base pairing. K+ stabilizes specific as well as non-specific annealing. The bases from opposite strands anneal due to hydrogen bonding forces. The better the base pair match of the two strands, the stronger the binding of the primer to the template DNA. Base pair mismatches result in binding forces that are not as strong and can be further weakened by ammonium ions. In an aqueous solution under thermal cycling conditions, NH4+ exists both as the ammonium ion and as ammonia, and interacts with the hydrogen bonds between the bases. The free electron pair of the ammonia molecule competes with the bases for the hydrogen in the hydrogen bonds and destabilizes base pair binding. Because hydrogen bonds are weak, base pair mismatches break and primers are then completely removed from the template strand. Only specific binding and perfect base pair matches resist the destabilizing effects of the NH4+ ions. Those primers with a perfect base match stay annealed and are preferentially elongated later on in the PCR process. QIAGEN's dual cationic formulation of K+ and NH4+ ions results in preferentially amplified specific products. Standard PCR buffers that do not contain ammonium ions stabilize both specific and non-specific primer annealing. Due to the dual cationic effect of QIAGEN's PCR buffer, annealing efficiency is only high with a 100% base pair match between the primer and the template. This effect and the resulting specificity is also independent of the annealing temperature chosen. The stabilizing-destabilizing effect of the dual cationic formulation of QIAGEN's PCR buffer system provides several unique advantages to researchers and increases their experimental success rate. This means no need for gradient PCR, no need for optimization of magnesium concentrations, a strong increase in specificity, one PCR protocol for all assays, time and cost savings, and ease of use. QIAGEN delivers PCR success at the first attempt.
QIAGEN.
Sample to inside.